Well, thank you for the introduction, and I'd like to thank the organizer for giving me the opportunity to talk to you all. I joined UCF in 2019, so I got the joy of setting up the lab during COVID. I've published about 17 papers since then. I teach Introduction to Pharmacology with Health Sciences courses, and I teach it in the med school in the foundations courses that they do in the first 2 years. The primary goals in my lab is to look at cartilage to understand osteoarthritis, primarily osteoarthritis, and we do that by engineering the cartilage. We also are looking at making peptides that will target the injured joint. The idea is there that we will find a peptide that will target the injured joint, which will enable us to home either a theranostic, so something that will tell you if somebody has arthritis, or actually a drug to treat it. We also are looking at making the tissue engineered so it could replace the cartilage, but the main focus of the talk today that I'll be giving is on using the engineered cartilage as a model for drug discovery. We go all the way from kind of the molecular level all the way through the cell and tissue level up to the whole joint and the whole body. The thing about before I actually get into that, the first thing I want to do is acknowledge the people that have done most of the work. So my lab has been made up primarily of undergrads and med students, shown in yellow and blue, with postdocs in pink and grad students in green. So Carrie and Rachel have actually spanned 2 of those positions, and then Nanya is actually a high school student. So you came in and did some of the genotyping work that we've done. Most of the work that I'll be telling you about today was done by my postdoc and grad student Carrie and Dr. Cruz. The work could not happen without the tissue, and the tissue comes from cells, and I brought cells with me. Cells have traveled all over the country, from Case Western, Baylor College of Medicine in Houston, and now I'm getting tissue from the International Institute for Advancement in Medicine and the Gift of Hope program and the Moors here in Orlando. We could, of course, not do any of this research if it was not for the money. So big thanks goes to UCF and UCF College of Medicine, the Rowland and Bird and Lawrence Bone Disease Program of Texas, and now to the NIH. So what is it that we're actually interested in? Well, as I said, I'm interested in osteoarthritis. The current status of arthritis is that most people, if you are over 65, about 50% of people get arthritis. And the problem is that the drugs are palliative. That means they're treating the symptoms, not the actual disease. Even with the stem cell injections, we're not seeing a clear benefit there, and it's very variable depending on where you get those cells from. So eventually you put up with the pain until you get a total joint replacement. And total joints are fantastic, but they have a limited lifetime of 15 to 20 years. They are infection prone, and you don't get the same functionality that you get if you actually had a functional joint. So cartilage is mostly extracellular matrix, and what if we could actually stimulate that cartilage to regenerate or prevent it from deteriorating? So that's where this really came as a push to me. It was I have family and friends with arthritis, and just seeing that there isn't anything to treat it just kind of stuck with me as that's either an opportunity or a hole that I'm just going to keep digging. So current drug discovery methods primarily use a target. So you need to have a receptor or an enzyme that you can target. And then you can use the in silico drug screening, or actually in vitro assays where you have a library of drugs or chemicals and see if they will bind to that protein. But cartilage is mostly extracellular matrix, and that's what your cells kind of spit out. So your cells are secreting this extracellular matrix, and it's that matrix that makes your cartilage, makes it work. And if you lose it, then you get arthritis. So to look at molecules or methods to stimulate the extracellular matrix, we focusing on a phenotypic screen. So that is screens that will show us that you are getting the tissue. The main problem with phenotypic screens is that assessing the extracellular matrix is time consuming, laborious, expensive. So that stimulated me to look into other methods of assessing this. And we landed on the Gaussian luciferase. So you've probably seen fireflies. There are marine copepods called Gaussian luciferase, and they secrete this luciferase and it actually distracts the predators. So it gives flashes of light. So what we did was take the protein that codes for the Gaussian luciferase and drive that with a type 2 collagen promoter. So type 2 collagen is the main extracellular matrix component of your cartilage. And then when we put that into the cells, so we're engineering chondrocytes, we have the type 2 collagen promoter driving the luciferase. The luciferase is secreted into the media that we grow the cells in. And then we can take that media, add the substrate, and we have an emission of light, and that emission of light correlates with the amount of the protein that we're trying to target. So there are several benefits of this luciferase. It is secreted. It is a non-destructive assay because it's secreted. It is both fast and sensitive. The substrate is cheap. And it means that we can take cartilage and we can grow pieces of cartilage in the dish and analyze them over time. So we get a temporal assessment of how we're affecting that extracellular matrix production. So typically in the lab, what we'll do is to take a vial of cells out of liquid nitrogen, so cryogenically stored cells, and we'll thaw them. So we thaw about 1 million cells, and we seed them onto a flask. So this flask has media that contains nutrients. So it's kind of like Gatorade for cells, maybe better. And the cells will multiply. So we go from about 1 million cells to about 20 to 30 million cells. And then we trypsinize those cells. So that means we dissociate those cells from the flask into a single cell suspension, and we make the cells into little aggregates of cartilage. So that's us putting them in these little drops, and then we'll centrifuge this down, and it makes a little ball of cells. And you can see as we analyze that over time, the blue dye here is staining the nuclei, and over this 10-hour period that we image the cartilage pellet, it contracts into this little ball of cartilage. And that's basically our testing method. So we take that ball of cartilage and we treat it with various different compounds. In this screen, we looked at a natural product library. The natural product library is about 400 different compounds, and we stimulated that over 3 weeks. So we would feed the cells every 2 to 3 days, image the little cartilage pellets, and analyze the amount of luciferase that's been released. So this is kind of the bioluminescent data. So as the cells secrete the luciferase into the media, we add the substrate and you get a level of bioluminescence. So on the y-axis is that luminescence, and on the x-axis we have the days in culture. So we sampled the media on day 1, day 3, as you can see these various different time points, and each line represents a different compound. The DMSO is a kind of vehicle control. So that's what you dissolve the natural products in to be able to put them onto the cells. So that's where kind of normal is. And then if we just didn't treat them with anything, you can see that overlays with that DMSO control very nicely. So the vehicle isn't having any effect on those cells. But, you know, kind of getting rid of all of this mess and looking at the ones that actually stimulated something, we can see we have deserpidine and aromaline here that were coming up at the end of this assay as stimulating the type 2 collagen. And these are two natural products, and we don't really know what those natural products targeted. So the next step was to look at target identification, and we did that in silico. So first we would put the structure into this in silico analysis, and it gives us these various different targets. And what came up were these dopamine receptors. And dopamine is an interesting target because your cartilage doesn't have any nerves in it. So this was kind of a surprise. But then when we looked at some RNA analysis, so that's what your chondrocytes are actually producing, the proteins that they're producing. The only dopamine receptor that they were producing was this dopamine receptor D4. And, you know, that then kind of stimulated us to then go look at dopamine receptor agonists and antagonists, and we found that an agonist at the dopamine receptor gave us similar data to what we were seeing with the aromaline. And, you know, if you want to kind of look at that more in depth, I invite you to go look at this paper. So we published this about a year ago, and this kind of describes the process of making the cells, validating those cells, and then screening all of those compounds, and then validating the compounds that actually came up as a potential hit. So now we found a target. So this then led us on to looking into the effect of the dopamine receptor D4 knockout. And there were no skeletal phenotypes reported in the literature. So we thought, okay, well, what if we disrupt the system? So what if we cause an osteoarthritis model? So we take the knockout mouse and we put it into this non-invasive compression device, and that compresses the knee and gives it an ACL tear. So kind of like people, sports injury. Unfortunately, a lot of people that get ACL tears will go on to have post-traumatic osteoarthritis. And then we look at the mice and we look at them in terms of pain, gait, so how they're running on a treadmill. We have in vivo matrix metalloproteinase. So matrix metalloproteinase is the enzymes that digest the cartilage. And then at the end of the 8-week course, we were looking at the micro CT, and here we can look at the medial and lateral compartments of the joint, and you can see that we have a significant increase in the injured leg over the uninjured leg. A significant decrease in the bone volume in the injured leg over the uninjured leg, and that's both in the medial and the lateral for the knockout, but not in the wild type. And we see some various different disruption of the joint with the disease. So that's kind of where we are right now. Looking forward to getting this published. We have, you know, we go all the way from the molecular to the whole body, so we have lots of opportunities for collaboration with material science, computing, you name it.